Hey all, welcome to ShareTreck. This is Raj here. Friends, today I'm going to talk about a concept that would be, I think, very nice for any genomic investor to have in mind when they look at their investments and future trends. And this is going to be a comparison between ADC and CAR-T. ADC is the antibody-drug conjugate. So in this video, what you will get to understand is what is ADC, how does it work, and what is it effective on at this point of time. I will probably give you a couple of examples of already approved ADCs which are doing well in the market. And then I'm going to compare and contrast them with CAR-T and going to tell you why ADC and CAR-T have not yet clashed as much right now and what the future holds and what does CAR-T have to do in order to compete with ADC. So those are the things that I'm going to talk about in this video. With that said, let's get started. Welcome back, friends. ADC or antibody-drug conjugate and CAR-T or chimeric antigen receptor T-cell are both promising approaches in cancer treatment. ADC involves linking an antibody to a chemotherapy drug. The antibody targets specific proteins on cancer cells, delivering the drug directly to them. This minimizes damages to healthy cells. And CAR-T, on the other hand, is a type of immunotherapy. It involves modifying a patient's T cell or a donor's T cell to express a receptor, which is chimeric antigen receptor, and hence the name CAR-T, that recognizes and attacks cancer cells. Monoclonal antibody are designed to target specific antigens and that are present on the surface of cancer cells. These antigens are often proteins that are overexpressed in cancer cells compared to normal cells. The selection of the right antibody is crucial as it determines the specificity of the ADC for the targeted cancer cells. So the ADC basically has an engine driver, which is a monoclonal antibody, and that antibody has a propensity to adhere to a specific antigen, and that antigen has to be prolific on the targeted cancer cells in order to make the ADC more effective. In an ADC, apart from the antibody, there is a linker molecule. The linker molecule serves as the bridge connecting the antibody to the chemotherapy payload. There are two main types of linkers, cleavable and non-cleavable. Cleavable linkers are linkers that can be broken inside the cancer cell, releasing the chemotherapy drug. This can happen through enzymatic processes or changes in cellular environment. Non-cleavable linkers are more stable and release the drug only when the entire ADC is internalized by the cancer cell, and the chemotherapy payload is basically the warhead that is detonated inside the cancer cell by the ADC. It's a potent cytotoxic agent designed to kill the cancer cell upon release. The choice of chemotherapy drug depends on the type of cancer being targeted and its sensitivity to specific drugs. Example of specific chemotherapy payload include MMAE, which is monomethyl auristatin E, used in Kadcyla, which is another drug, and MMAF or monomethyl auristatin F, used in Adcetris, which is another drug. So the key innovation with ADCs is the ability to deliver the potent chemotherapy directly to cancer cells, minimizing damages to healthy tissues and reducing systemic side effects, thereby improving patient outcomes as well as patient experience. Antibody-drug conjugates have been in development for several decades, but their significant clinical use started gaining traction more recently. The first ADC to receive regulatory approval was Mylotarg, and that was in 2000 for the treatment of acute myeloid leukemia. However, due to concerns about safety and efficacy, Mylotarg was withdrawn from the market in 2010. The field saw a resurgence with the approval of Adcetris in 2011 for the treatment of Hodgkin's lymphoma and anaplastic large cell lymphoma. Since then, several ADCs have been developed and approved for various cancers. The development and refinement of ADC technology continue with ongoing research and clinical trials exploring new targets, linkers, and payloads to improve efficacy and reduce side effects. Companies like Genmab, Roche, Gilead Sciences, Immunogen, and Seattle Genetics have been leading with their work on ADCs. However, does this matter to companies in our watch list? That's the main question I want to answer today. Well, CAR-T has been very successful with blood cancers, where ADCs have been more successful with solid tumors. So any genomic company creating CAR-T therapies will find that one of the reasons why CAR-T seem to be less successful with solid tumors is that solid tumors create an environment that subdues the activity of the T cell. We had discussed PD-L1 situation earlier as an example. So there is a need for an extra layer of work to be done for a CAR-T therapy to be successful with a tumor. So that adds more complications. And again, if the CAR-T is from the same patient, it is much easier. If it's a donor CAR-T for a allogenic CAR-T therapy, then that becomes even that much more difficult. That said, I thought it would be good for genomic investors to understand these nuances. And now with this knowledge, if we were to look at all the pipelines of all the genomic therapy companies in our watch list, then we would find that there are certain companies which are trying to create a CAR-T therapy to target solid tumors. Those are the ones that are going to compete with ADCs. And if they happen to be autologous, then all the demerits of autologous accrue to it. And if they are allogenic, then the benefits of allogenic therapy accrue to it, but they follow they will have a lot of other hurdles to overcome in terms of graft versus host disease and various other constraints. And over and above this, they will have to do something about PD-L1 because that's what many cancer tumors use in order to subdue the T cells in their vicinity and continue to evade immune system. So that's my basic understanding. So maybe in the future we'll have situations where ADCs and CAR-T might probably compete directly, but at this point of time, I don't see a competition or a major overlap. So with that, my friends, I would like to end this video. This was a little bit of a knowledge bite, and when I started reading about ADCs and CAR-T, and things started forming in my mind, the definition and the understanding started forming in my mind, I thought this would be a nice bit of information that our viewers here in ShareTreck would like to have and keep it handy for future use. With that, my friends, I would like to end this video here. If I find more information and more clarifications, I would probably produce a sequel and give you more clarification. With that said, I'll catch up with you in the next video. Bye for now.